Good evening, everyone, and thank you for joining us in our edition today of Brain Bridge. This is our webinar series that we organize every other month, where we invite experts from around the world to share with us cutting-edge research in their respective fields. Our speaker today is Dr. Ralda Nehme from the Stanley Center for Psychiatric Research, where she is the Institute Scientist and Director of the Stem Cell Program. Her research is focused on the way brain cells talk to each other, how genomic variants shape that conversation, and how stem cells can inform us better on the conversations between brain cells and why it matters for mental illnesses like schizophrenia. Before I turn it over to her, just a couple requests to everyone joining us today. Please keep your systems on mute so that the presentation can go smoothly. We'll have a Q&A session after the presentation, and welcome to type your comments and questions in the chat box at any point, and we'll take them up at the end. Thank you so much again for joining us, everyone, and Dr. Ralda, over to you.
Thank you so much, Sharika, for this kind introduction, and really also for the whole group for the invitation to speak here today. I'm really pleased to be here and have this opportunity to share our work, and I look forward to the Q&A with everyone as well. So as Rilika just said, I would like to take this opportunity to really kind of highlight how our approach using human cellular models that are genetically diverse can help us really get some insight into what, the cellular and molecular mechanisms of psychiatric disorders. So as this audience is undoubtedly well familiar with, mental illness continues to be a major global health challenge. And this is because these illnesses are usually linked to severe symptoms, from mood swings to hallucinations and an increased risk of suicide. They also affect millions around the world. They contribute to homelessness, unemployment, and incarceration. And notably, the treatments that are now available are actually outdated. There has not been, for most of these conditions, there have not been new treatments that have come to the market that are actually effective at treating the majority of the symptoms. And over half of the people that have these conditions still struggle with daily functioning. So this is all to say that we need to, to keep working very hard to understand, what's going on at the mechanistic level, and really with a focus to developing new therapies. The good news is that human genetic studies over the last decade or two have uncovered hundreds of gene variants that are linked to psychiatric conditions, such as schizophrenia, for example. And again, we all know that these conditions are highly complex and heterogeneous, and this is true both at the clinical level with the diversity of symptoms that are linked to these conditions, but also at the genetic level. So there's not only one genetic variant, but many, the rare variants, the common variants, and hundreds of them that are linked to these conditions. And so what my lab, and I'm sure many others as well, including in this community, want to know is, what are the gene variants doing? How these genetic variants affect and shape cellular programs, and then in turn, how this affects brain function and development, and how when these are not functioning properly, then it would result in disease in some individuals. So essentially, we really want to bridge the knowledge gap between what the gene variants are and then what their function is, and eventually leading to improved therapeutic discovery. And ideally, to understand that, we would need to be able to study and manipulate the living human brain. However, for many different reasons, this is obviously not ethical, but it's also not possible. We can't really access the human brain at many different stages of development, right? There's obviously human postmortem studies are really powerful, but again, it's just they're limited in what we can do with the tissue that is available. So for these reasons, we turn to human pluripotent stem cells because they give access to the tissues that are actually affected in these conditions, but also very importantly, they preserve the whole genetic background of the individual that the cells were derived from. So we can derive human stem cells from any given individual anywhere in the world. for example, by taking a blood sample, a skin biopsy. Then once we have these tissues, we can turn them into stem cells in around like a few weeks. And then once we have the induced pluripotent stem cells, then we can turn them into any cell type we're interested in. You know, you hear me talking a lot today about neuronal progenitor cells, neurons, and astrocytes. And again, these derivatives of the original primary tissue will have exactly the genetic background that was in the individual that they were derived from. So today I will share some of our work on establishing tools and resources to enable the kind of approaches that are going on now in the lab. Then I'll focus on a few vignettes to highlight how we implement these resources to study psychiatric conditions. I'll talk about our work on astrocyte and neuron interactions, and then I'll talk about our work on how these cells respond to perturbations of their biology, specifically how genetic risk variants may perturb this biology, but also chemical perturbations such as antipsychotic medications and cytokines. All right, so over the last several years, pretty much since we've set up the lab here several years ago, the first thing we realized is that it was not sufficient to just examine stem cell lines from a few patients and a few controls. So because of this heterogeneity, we just would not have the statistical power to make any meaningful discoveries that would rise beyond the noise of technical variability and biological variability. So for this reason, we really put in a lot of effort, and that was a large collaborative effort to assemble stem cell lines from many different people, again, all around the world. And for each of these stem cell lines, we wanted to obviously QC them and bank them, make sure that the stem cells are really pluripotent, that they are not don't have aberrant differentiation, et cetera. But we also made sure to have whole genome sequencing data and SNP array data from each and every cell line, again, that we will then leverage in downstream experiments. So all in all, we were able to assemble a collection of stem cells from over 1,000 unique donors. And we also then developed approaches to be able to turn these stem cells into the cell types we're interested in a manner that was really scalable and as little heterogeneous as possible. So we now have approaches to be able to make homogeneous preparations of someone is requesting remote control of my computer. Should I accept or decline?
No, please decline. Yeah.
Okay. All right. So then now that we have these homogeneous preparations of neural progenitor cells, neurons, and astrocytes that we've been able to generate from hundreds of stem cell lines, now we were well poised to, again, examine what the specific variants were doing. And then last, I'll highlight one kind of approach that really also enabled us to even further increase the scale of the experiments that we could do is that, and this is a pooled approach, so which we call cell villages. This was developed together with Steve McCarroll here at the Broad, who's a human geneticist. And so in this approach, we mix different cells from many different people all together in one dish. And then we take advantage of the fact that each one of us come pretty much endogenously barcoded, and that we all have unique single nucleotide polymorphisms in our genomes. And then these SNPs can serve as a barcode that basically then will help us decode the donor of origin for each cell following a single cell RNA-seq experiment. So this cell village experiment obviously has its limitations in that we can examine mostly the cell intrinsic or non-cell autonomous effects. But again, in some cases, it can really help increase the scale at which we can do these experiments. And we actually, over the last couple of years, we've been able really to, somebody called it tame the cell villages, and that when we first started working with these cell villages, we noticed that when cells, proliferative cell types, mitotic cell types, such as stem cells, when we mix them all together, we noticed that one or a few cell lines would quickly take over the cultures. These, after we further examined these cell lines, we noticed that many times these cell lines would have some karyotypic abnormalities that we then needed to exclude them from cell villages. We also noticed that different cell lines, they grow at different rates. So we should be mindful in kind of mixing together cell lines that grow at at similar rate. And we also noticed that we should really avoid passaging cells as much as possible before starting an experiment after we mix them, because each time we lift the cells off the plate and then plate them again, different cell lines would adhere to the substrate, to the plate at different rates. So this is all to say that, we have been able to overcome these hurdles, and now we can generate stem cell villages from 40 to 50 people at once that really over several days in culture can maintain a stable representation of donors. Obviously, this has limits. We can't passage them for weeks. So we try to just keep them together for no longer than a week and then put them in an experiment. All right, we also have been able to freeze these cell villages. So we make a stem cell village, we freeze it, and then we induce neuronal differentiation, again, all the while preserving a stable representation of donors in a village. Again, these are all approaches that enable us to move closer to a kind of population scale of experiments in the dish. All right, so how can we then use these tools and resources to understand the complexity of human traits and disorders? And so I'll, for the rest of the talk, I'll focus on site interactions, which I'll start with, and then again, the perturbation of the system using either genetic risk variants or chemical perturbations. So if we think about kind of the human genetic studies and the genes and pathways that have been implicated in schizophrenia and other psychiatric conditions, synaptic transmission comes to mind first and foremost. And when we think about synapses, we often think about connections or neurons, or connections between two different neurons, a presynaptic neuron and then a postsynaptic neuron. And so this has been extensively studied. The risk variants have been often studied in the context of such neurons, again, whether there's a presynaptic role or phenotype or a postsynaptic role and phenotype. But for a while, the field has overlooked that there is actually another cell type that is a very important component of synapses, and that's the astrocyte cell types. And so we wondered if other cell types, such as astrocytes, could also be vulnerable to these kind of changes, like to the variation, genetic variation that is linked to psychiatric conditions. And just kind of, again, to bring everybody up to speed here about astrocytes. So astrocytes are a key component of the synapses. They are kind of essential for proper neuronal development and maturation. So they have a structural role at the synapses, again, engulfing the synapses, but again, also a very important functional role to kind of enable neurons to really achieve their proper maturation. So we wondered if neuron and astrocyte interactions may change in psychiatric conditions. And to be able to look at astrocyte-neuron interactions in the dish, we compared cells that were grown together in co-cultures, so neurons and astrocytes grown together in co-cultures versus neurons and astrocytes that were grown separately, each in their own dishes. And so all in all, we studied cells from over 80 individuals in this study. And what we have been able to find is that actually astrocyte and neuron gene expression programs change dramatically whether cells are grown separately or together. When we put the two cell types together in co-culture, we noticed that, perhaps not surprisingly, synaptic genes increase in their expression in neurons. But also we noticed that when we looked at changes in the astrocyte cell type, that cell adhesion genes also increase in astrocytes. And perhaps that was the most surprising for us, is that also cholesterol production genes dramatically increased in astrocytes as well. And here I'm highlighting an example of two such genes in astrocytes. The first is Neurexin-1, which is classically studied as a presynaptic cell adhesion genes. And it's also a schizophrenia and an autism high confidence risk gene, again, studied extensively in the context of neurons, but here we show that Neurexin-1 increases dramatically upon co-culture. And the second gene is HMGCR. This is a rate-limiting enzyme in cholesterol production, and it also increases in co-culture. And kind of very interestingly, soon after we had made these observations, a study from our colleague here at the Broad, Steve McCarroll, led by postdoc Emmy Ling in his lab, they also found that actually these same pathways are also changing in a coordinated manner in postmortem data. And when they studied postmortem brain tissue from roughly 200 people, half with and half without schizophrenia, they noticed that the people who have more expression of synaptic genes in their neurons also had higher expression of cholesterol and cell adhesion genes in their astrocytes, and that both of these were reduced in individuals with schizophrenia. So they called this synaptic neuron astrocyte program. And then again, they showed that it was reduced in the brains of schizophrenia individuals. So then in a follow-up study, we actually wanted to examine more deeply the dynamics of these changes. And so we conducted a kind of a time point study and a transcriptional study, transcriptional profiling studies, and really with a focus on the astrocyte cell type this time. And we found that actually almost immediately after we put the neurons and astrocytes together in a dish, so immediately after they make physical contact with each other, we have the cholesterol biosynthesis genes that are induced. So this happens sometime between 6 hours and 1 day. So some genes kind of come up earlier and some genes later in the cholesterol pathway. And then also the cell adhesion genes also come up within a day after co-culture. So very, very quick changes here. And again, many of the genes here are linked to schizophrenia. And so this is perhaps some people may wonder, how come the cholesterol biosynthesis pathway? So what is it actually, did it come up in the genetic studies? First of all, if we kind of think about synapses, cholesterol is really an important component of cell membranes. And it turns out that neurons rely on astrocytes to synthesize cholesterol, and then they import it, and then it becomes a component of the cell membranes, which obviously is a really important component of synapses. So it makes sense that this link between cholesterol production and synapses is there. And when we then lay out the cholesterol production genes and the cholesterol uptake genes on the neurons, so cholesterol production in astrocytes, so such as the SHRB F1 and F2 transcription factors, the cholesterol synthesis genes, HMGCR, and the lipoproteins via which cholesterol is packaged once it's secreted outside astrocytes and then taken up by neurons using the receptors LRP1 and the LDL receptors. So it turns out that many of these loci, all of the ones highlighted here in blue, actually happen to be near loci that are associated with schizophrenia. So we think that these findings are actually consistent with the human genetic studies. All right, so from there we wondered, well, what happens if we then prevent neurons from taking up the cholesterol that's being produced by astrocytes? So we did this by using CRISPR-I to downregulate, to knock down the expression of LRP1 and LDLR, the cholesterol biosynthesis genes. And when we do that, we found, like as shown here in this heat map, that the same synaptic genes that are normally induced in neurons upon physical contact with astrocytes, so these are the genes highlighted here, so these genes are then decreased in their expression when we prevent neurons from taking up cholesterol produced by astrocytes. Again, further strengthening the link between these two pathways and the coordination across cell types. So from there, our next question was, well, do genetic risk variants affect neuron-astrocyte interactions? And so I'm going to highlight here our work on one genetic variant, but again, we're starting to look systematically at many of the genetic variants that have been implicated in schizophrenia. But one of the risk variants that we have been studying for a while now it's a rare variant. It's the 22q11.2 deletion, which is actually one of the most common chromosomal deletion in humans. And then so this deletion takes up roughly 50 genes on the Q arm of chromosome 22. So we have assembled a collection of people, case control cohort, that either have or do not have the deletion. We also, using CRISPR, have removed this deletion from an otherwise neutral genetic background. And so, and then we've performed transcriptional profiling on initially the neuronal cell types and then the astrocytes more recently. And what we found is that upon deletion, heterozygous deletion of this region, we can observe cell type specific gene expression changes. So we observed that in neurons, when this deletion is region is missing, we observe that synaptic genes that have been implicated in schizophrenia are decreased in their expression in neurons with 22q11.2 deletion. As you can see here, we also validated these findings by immunostaining and proteomics. And then more recently, we looked at gene expression changes in astrocytes with this deletion, and we found that the astrocyte component of the synaptic neuron astrocyte program is decreased in neurons with this deletion. So again, indicating that this, the SNAP program in neurons and astrocytes overall is kind of also indeed decreased in neural cells from individuals with this deletion. So we also wanted to kind of beyond gene expression changes, we also wanted to study genetic influences on kind of other levels in the cells. So we focused on cell morphology, which is a readout we've become increasingly interested in over the last several years because of advance in basically multiplexed immunofluorescence approach that enable us to capture a lot of data. And then also methods to analyze these images that are really deep learning and AI based. So we, in an initial study, we used the cell painting assay, which uses 6 different dyes to label different cell compartments and organelles in the cells, such as nucleus, cytoplasm, mitochondria, ER, the cell membrane, etc. So we can label all of them with very cheap dyes. And then from a single image, we can extract thousands of features then that enable us to then we can then analyze this data similarly to how we analyze RNA sequencing data. So in initial study, we profiled induced pluripotent stem cell lines from roughly 300 people. And then we, because we also have the genotype data, the whole genome sequencing data from each and every person, we were able to run basically cell morphology quantitative trait loci study, whereby we could link a specific genotype with a specific cell morphological trait. And then, but we had done the study just on stem cell lines, to be able to scale in an arrayed format, non-pooled format. And then in a subsequent study, we then applied this assay to neural cell types. So we developed neuropainting, where we applied cell painting. We developed slightly different kind of wet lab, but also analytical pipelines to then profile neuronal progenitor cells, neurons, and astrocytes. And we applied neuropainting to our case control cohort with 22q11.2 deletion. And so we were able here again to see that we can detect cell type specific changes in morphology that are linked to the deletion. And so we identified many different significant features in stem cells and neuronal progenitor cells and neurons and astrocytes. And you can see that while some features are changed in all cell types, there are many morphological traits that were uniquely changed in each cell type. And for example, here I'm showing you the heat map for astrocytes with or without the deletion that you can see here. So the green indicates cell lines that don't have the deletion, and purple cell lines with the deletion here in this heat map. And again, we identify different morphological traits that are changed based on the deletion. For example, here you can see that the distribution of the ER and the mitochondria in cells with or without the deletion, it's different. So many of the morphological traits actually that come up as differentially expressed are ones that it's very hard for the human eye to see and interpret. But this is one that actually we can see and interpret. And so this is why I'm highlighting here that you can really see that there are differences here. And then actually more recently, based on our findings on neuron and astrocyte interactions, we started becoming interested in lipid profiling. And then more recently, and this is work by Jack Su and Emily Henson in the lab, we've implemented, we added a lipid dye to our neuropainting panel. And so this is unpublished still, but it was kind of really interesting to see. So this is the astrocyte cell type again. It was really interesting to see that actually We had here again a significant difference in cells with or without the deletion, and the number of spots per cells, but also the area, how big the area of the lipid droplets were in each cell. And what I'm not showing here is that actually from the lipid features alone, we can predict whether a cells may or may not have the deletion, basically. So that was really kind of like interesting to us that we have a lot to gain by adding this dye. And again, from the morphological features alone, but even the lipid features specifically, it's they are sufficient to separate the deletion cells from the controls. We also then went on to develop a FACS assay, so we can use this dye and FACS astrocytes that have a high lipid content versus a low lipid content, and you know, we can by just going to the tails, we can pretty much separate out cells with or without the deletion. So from there, we wanted to basically ask how these astrocyte neuron interactions might respond to perturbations of their biology, chemical perturbations of their biology. And so in this experiment, we really kind of wanted to take a more population scale functional genomics approach. So we took a cell village of 140 donors and then applied 20 different treatment conditions. So that was really an experiment that was done experimentally. It took one graduate student a little over a month to complete this experiment. So we took here neurons and astrocytes, applied all of these different treatments here, and then performed single-cell RNA sequencing. The analysis of this data set obviously took a long time, but you know, again, it shows the power of just multiplexing and scaling up in this format. And so today I'm going to focus on two vignettes here. I'm going to tell you about our findings of antipsychotic medications and also on cytokine treatment. So again, thinking about antipsychotic medications, this work was led by a really talented graduate student, former graduate student in the lab, Matt Techmeyer, who has now started his own lab at Purdue University. And so what we found there is that upon treatment with the antipsychotic clozapine, we can see a really robust induction in genes with functions in cholesterol biosynthesis and transport. And so this was really very robust. It was really pretty much unique to clozapine. We found that this was true not only in cells from one given donor, but in each and every individual that we examined, we could see an increase in cholesterol biosynthesis genes upon clozapine treatment. And we then went on to validate this at many different levels using qPCR, also by measuring total cholesterol inside the cells, also measuring the cholesterol that was secreted outside the cells and the supernatant, as well as the cholesterol that was packaged in the apolipoproteins, ApoE and clusterin. So again, throughout all over the board, we see that we could always detect an increase in cholesterol production at all of these levels upon clozapine treatment. So really very robust signature. And so clozapine, as many here might know, is a particularly effective antipsychotic medication, but we don't know why it works so well. So we wondered if some of its therapeutic benefit may be from effect on cholesterol synthesis. And so then we went back to our system and looked at, in a co-culture system, looked at astrocyte and neuron co-cultures that were treated with clozapine. And here we could see that indeed, in a dose-dependent way, we could detect upregulation of cholesterol biosynthesis in astrocytes. The cell adhesion genes were also increased. And then when we looked at the co-cultured neurons, we then could detect a really nice, again, dose-dependent increase in the expression of synaptic genes. So this was done using kind of latent factor analysis approach. So again, this shows that this, again, SNAP pathway that is ordinarily decreased and individuals with schizophrenia can be now increased using clozapine. And finally, we also looked at cell morphology. I'm not showing this data here, but just to summarize, the top 50 differential features was, so when we look at astrocytes with or without 22q11.2 deletion, for example, we see that there are 50 morphological features that are significantly different in astrocytes here. But however, these features, when we treat clozapine, so the difference between cases and controls, so becomes more subdued. So clozapine at least partially rescues morphological differences linked to 22q11.2 deletion in astrocytes. All right, so then now I'm going to switch gears a little bit and kind of share our work on, again, using the same system, the cell village approach, but now neurons treated with cytokines. Our question there was, how does common human genetic variation shape the response of neuron to inflammation? We know that different people respond differently to neuroinflammation in the context of, like disease development, but also trajectory. It can be very different from one person to another. And this work was led by Milena Enzelm, who's a physician scientist, was a postdoc in the lab, again, now also started her own lab at Boston Children's Hospital. And Milena is a pediatric neuroimmunologist in the clinic. So she sees a lot of kids with these neuroimmune diseases and kind of was always perplexed by why, like the presentation is so different across different patients. And Milena was joined by Sonia Bolshakova, who's a computational associate in the lab, who helped a lot with the analysis. So again, this is our village of 140 donors, and here we're treating the neurons with different cytokines. We're gonna talk mostly today about interferon alpha and interferon gamma. And so the first thing that we found here is that looking across the board is that we could detect many gene expression changes that are induced by interferon treatment. So a lot of the genes that we expected to be there come up. So very robust response, very reproducible response for both alpha and gamma treatments. However, what we found was that while all donor neurons respond globally to interferon treatment, we found that there were gene expression differences across the donors. So again, here you can see that all donor neurons, so each dot here is a different donor. So all the neurons respond to interferon gamma and alpha treatment. However, if we look at the genes that are induced by interferon alpha, for example, so each row here is cells from a different donor, and then we have the induced genes that are here represented. And so you can see that there is incredible modularity in this response. So in some donors, we have some genes that are like downregulated in some donors, but they are robustly expressed in other donors. So the same genes that are, again, induced here across the board. And so from there, again, to focus on natural variation that is genetic in origin. So again, variation in how cells from different people respond to cytokine treatment could be too many different causes. What we can really capture nicely in our system, variation that is natural and genetic in origin. So to get to this, we wanted to examine expression quantitative trait loci. So these are gene expression changes that are due to single nucleotide polymorphisms at specific loci. So we here examined such EQTLs. We found that we had EQTLs that are shared across all different conditions. So baseline interferon alpha, interferon gamma treatment. So many changes in gene expression we could attribute to specific nucleotide changes already at baseline. However, we had also many that were condition specific to each condition, many EQTLs that were specific to either treatment with interferon alpha or gamma, for example. For example, here I'm highlighting a change in gene expression in AgTrap. This is the AgTrap loci or gene. You could see that how, we don't see a change in gene expression in the control condition or the interferon gamma condition, but very robust genotype-dependent change upon interferon alpha treatment here. And then for another gene, caspase 7, we could see a really kind of robust interferon gamma dependent change here, and then some EQTLs that were present across all conditions. All right, so how do we then make sense? I just showed you that we had like hundreds of EQTL that are changed. Not surprisingly, in our results as well, EQTLs are mostly intergenic or intronic. This is really consistent with all of the different broader EQTL studies in the field. So how do we then begin to annotate and interpret these changes and kind of try to start to put them together in a kind of kind of gain more mechanistic understanding. So looking at interferon alpha and interferon gamma, like the signaling between downstream both of these cytokines, we can see that there's one transcription factor, STAT1, that is kind of have been robustly and thoroughly studied downstream both interferon signaling cascades. And so we wanted to examine genome-wide STAT1 binding in neurons, so see which are the sites at which STAT1 binds in neurons. And we could do this using cut and tag. So this is basically a kind of newer version of ChIP-seq, where we could examine transcription factor binding in the genome using STAT1 antibody. And so Milena performed this experiment, and then we were able to detect really robustly that we could identify STAT1 peaks in the control condition and in the interferon alpha and gamma condition. So we detected increased STAT1 binding, again, consistent with our expectation, increased STAT1 binding in both the interferon alpha and interferon gamma conditions. And then what we found is that when we then overlaid on top of this information, the EQTL information that we had already, well, we found that when we looked specifically at the interferon responsive EQTLs, be it interferon gamma here in blue or interferon alpha here in red, we saw that we have, in each case, we had the interferon responsive EQTLs that were enriched in STAT1 binding. So again, leading us to conclude that STAT1 binding underlies much of this inter-individual variation in response to interferons. And then one example I'm highlighting here is finding on the Cas7 locus. So in this locus, we have, again, I had mentioned it briefly earlier, we had interferon gamma dependent EQTL at this locus where we only see an induction, genotype dependent induction upon interferon gamma treatment. We validated this at the protein level. And here we can see that whenever we have the minor allele, we can see increased STAT1 binding in the C allele. And the C allele was actually found to be protective against the development of Alzheimer's. It's actually present in 10% of people. And so we think that whenever we have here the C allele, the cells become better at completing apoptosis and leading to an overall decreased risk of Alzheimer's disease. So with this brings me to my concluding slide here. I told you today that astrocyte neuron interactions play previously unappreciated roles in psychiatric disorders. We think that the biological programs we discovered are consistent with human genetic studies, and the fact that we can manipulate these programs in living human cells and modulate them with drug treatment holds great promise for therapeutic discovery. And lastly, we found that STAT1 binding underlies much of the inter-individual variation in response to interferons. With this, I would like to thank all the individuals who've contributed samples to enable these studies, and of course their families. I'd like to thank everybody in my lab who actually led this work, our funding sources, and also collaborators. And thank you for your attention, and I'm happy to take questions now.
Thank you so much for the very exciting talk, Ralda. We do have some questions in the chat. I have one to start. So in the cell villages, do you have, because we're doing case control comparison here, is it one cell village for cases and one for controls, or they're both in the same?
Yeah, that's a great question. We actually mix them because we want to minimize batch effect. So we always try to have cases and controls in each village if we're doing a case control comparison, obviously. Yes.
So the data that you showed today was from a mix, like cases and control.
Yeah, exactly. Yeah.
So then that brings me to a second question then, because you're showing that there is gene expression differences when the cells are cultured together, which is, there has been evidence of that from a long time. So isn't there a concern that the expression of cases can affect the way controls behave and vice versa?
Yeah, again, so here in this specific study we're really focusing on the gene intrinsic pathways. So we also have, and I haven't shown that, we also have kind of single cell line, but also mix and match experiment where, for example, we have for the 22q11.2 deletion, we have astrocytes with the deletion and control neurons and vice versa. So we always complement these studies with arrayed
Okay.
cell line experiments. But so these gene expression programs here are in the cell villages. So not everything I talked about was in cell villages, like, but in the cell villages, we only focus at kind of cell intrinsic changes. Obviously, there is non-autonomous changes that we
Right, of course. Okay, so from the chat, we have one question from Parvati saying if you could elaborate on the inclusion criteria of stem cell branching process.
Yeah, so I think it's a very kind of intricate process. As you might know, we have to start with the consent forms, right? The patient consent forms. And there's a difference about how we were approaching it several years ago versus now how we would approach it. I think now we would just really only approach it pretty much hand in hand with the genetic studies, because we try to have, at the same time as individuals are being consented for a genetic study for a sample of DNA, we also ask if they would be okay to include, to have their stem cell line or just blood samples banked for stem cell derivation. Originally, they were a little bit, the earlier studies were separate, but so that's where we start, right? The consent forms, what, how broadly or not broadly consented. Some individuals want only to have their cell studies and for specific conditions versus broadly. We try to have them now be as broad as possible and individuals can opt out. But again, initially it was different. We also do a lot of, so we don't do the reprogramming ourselves. There's a wonderful stem cell core at Harvard that does the reprogramming for us. And they're, they do also really rigorous QC, but even after we get the cells, we also do SNP array data regularly, just to, because stem cell lines, as they are cultured, could acquire karyotypic abnormalities, and we don't really want it as a confounder in our study. We also run concordance analysis because we work with so many cell lines. It's really important to make sure that the cell that we think is from person A is really from that person. So we run like genetic concordance analysis to make sure that cell lines are what we think they are. Yeah.
Second question from Sudha Rao is, are organoid models tested? But I think what you showed today is only 2D.
It's everything that we showed is 2D. We have done some organoid work using also cell villages. That was mostly done in collaboration with Paula Arlotta. The scale, because organoids have different cell types, it's kind of much more complicated to make organoids, right? Like even there's a lot of cellular like division, like proliferation states that you can't really get around. So we found that for organoids, we can't really start with stem cell lines and then in a village and then make an organoid, because one or two cell lines again will take over. So there we had to push each cell line separately to become at the neuroepithelial stage, and then we can mix them together and make organoids. And also the scale by which we can do this is lower. So we could do, 5 to 10 robustly. If we stretch it a lot, maybe 20, but beyond this, it's not really sustainable to work with villages and organoid with this format. Obviously, it's still being developed and optimized, but it's possible, but none of the work that I showed you here is organoids.
Second question is from Parvati, is there any specific reason? I think this question pertains to the co-culture work of why the astrocytes are of mouse origin.
Yeah, this is also, for the sake of time, I didn't show you that work, but initially we started with mouse astrocytes just to look, because it was easy using bulk RNA sequencing to differentiate, to align to the mouse genome versus the human genome. So we could kind of maybe more cheaply and scalably examine expression changes across both species, but we have since also developed a fully human co-culture model, and the later experiments were actually done with this. And I didn't talk about the development and establishment of this model, but we are now basically on all of our co-culture experiments are fully human co-culture model. Yeah.
And this next question is pertains to the postmortem study you talked about.
Okay.
Bhagya's question is, which region of the postmortem brain was taken?
Yeah, so this was not our study. This is the work of Steve McCarroll. And so that was prefrontal cortex. But I know that they are now looking at many different regions. But that published work was from the human prefrontal cortex. Yeah.
And Did you also get to check if the cholesterol import genes are correspondingly upregulated in the neurons in the clozapine group? In addition to the astrocytes, I think are the neurons genes also affected?
I do not recall if that, but I can tell you if specifically the cholesterol import genes, but I know that just more generally, the cholesterol biosynthesis pathway is also increased in neurons. So that was not astrocyte specific phenotype. So when you treat with clozapine, it increases cholesterol production across the board.
And this is acute effect of clozapine, right? One day.
So 72 hours, I think
72
but we
hours,
also see it even in one day, even as quickly as like 90 minutes. So it's acute, but it then lasts.
Okay.
Yeah.
And do you expect to see clear signature if you ascertain polygenic scores as suggested before, or focusing on large effect variants? I guess because you talked about 22q, so if you're looking at PRS across groups where the individual effects are not so great, do you expect similar results?
So we haven't exactly looked again, and this was perhaps a matter of power. We haven't really examined PRS versus non-PRS. Again, this is because of the way the initial, like the stem cell lines were derived. So we don't have enough stem cell lines from the far end of the tails. But I can say that in a study that, we're publishing soon, actually, we're submitting soon, we have taken a village of 18 people and then introduced Neurexin 1 deletions and LRP1 deletions in individuals with high, low, and neutral polygenic risk score. So as opposed to comparing the polygenic risk score tails directly to each other, we compared the effect of the same mutation in different genetic background. And there we see an effect. But the most interesting thing is that we see an effect at baseline between high and low polygenic risk. We're very cautious with this because, again, our numbers are not very high, but we can see that in each donor, the gene expression changes, including in cholesterol, are different at baseline, and the effect of the mutation on top of that will make a big difference, but mostly because of the baseline gene expression changes are different. So
that is very interesting, and it makes sense that we would see that, considering that the effect of genomic background, which hasn't been explored so far. So it's
exactly,
very, very
yeah,
interesting
exactly,
that
yeah,
you're
yeah.
working on that. The next question is on if lipophilic statins would have any effect on cholesterol synthesis in astrocytes in this condition.
Yeah, so that's a great question. And we did look at that a little bit. We are still digging into this data, to be honest, more. We see at the gene expression level, when we added statin, we see that there's actually an effect on increasing cholesterol production overall. I should say at the gene expression level, right? Not at the, like when we haven't gone and measured cholesterol. But yeah, so the statins, because probably of feedback, there's a lot of feedback mechanisms in the cholesterol biosynthesis pathway. And so when we apply statins, the net result is that there's more gene expression increases, but we think that's actually, if we then go and measure cholesterol, they're going to be lower. So it's not as straightforward.
Right.
Yeah.
Right. Our next question is from Ria about the interaction that you've studied on astrocytes and neurons. And her question is if you could, what's your view on other kinds of glial cells and their interactions with neurons in similar experiments?
Yeah, so absolutely. And this is a direction that we're really interested in exploring in the future, either via like triculture models with astrocytes or even adding oligos or organoid models, right? We just wanted to because the nature of the question is so complex. There's so many gene expression changes, so many phenotypes we could be looking at. So we started like kind of in a very kind of reductionist way, exploring these, right? We started from the genetics. Okay, synaptic biology is implied. So we should start by looking at neurons, but also astrocytes are important. So we started from there, and I think are building up towards more complex systems because we think there will be kind of specific genetic variation that will influence cell type interactions between different cell types than just neurons and astrocytes. But that seemed to be, to us, the first place to start right now.
I think this is the last question we have is what would be the gene expressions result in prediction? And how will it emphasize the same on this? I'm not sure what, I think it's talking about the using AI for gene expression prediction differences.
Yeah, so I think we've used this mostly in the context of our cell morphology data. Now we're trying, and I don't know if I'm understanding the comment correctly, but we're trying to then go back and forth between gene expression and morphology. Can we predict one from the other, and what more can we gather by integrating both gene expression data and cell morphology data? And we think that there's actually a lot of benefit for the integration, and we can get to more biology, basically. Cell morphology data is much kind of cheaper and scalable to achieve, but sometimes it can be very hard to interpret. So what does it mean when whatever the texture of the ER is different between right cases and controls, right, biologically? So when we can lay on gene expression changes, then we can start to get to more biology that way, for example.
Right.
And also the other way around, there are some changes that we will miss at the right? Like, because gene expression is more upstream, there may be some phenotypes that will only, or some gene expression changes that won't lead to phenotypes downstream, right? So cell morphology is really quite a bit downstream. So it's very beneficial to use it to then predict which gene expression changes are the most impactful. So putting different modalities together is very powerful. And so, yeah, we're moving this direction.
Are you planning any MEA experiments to study circuit level changes?
Yeah, so we have as well. I didn't talk about it, but for the 22q project, for example, when it comes to studying single variants, we absolutely use MEA. We're also using optical electrophysiology, so this will be more compatible with the village format. But we've done so far a lot of MEA measurements, and we can validate the gene expression results with the MEA. Yeah.
All right, well, thank you so much. I think that's all the questions we have. I'll join echo the words of our audience to thank you very much. This was a great talk indeed, and was very helpful to know all the different directions that your work is proceeding. And we look forward to more interactions with you in future.
Thank you so much. Thanks for the invitation. It was really lovely, and thanks for the questions.